meiosis, actually part 2 of the lecture for 13. There's going to be a part 3 also. Okay, so we're on to the new topics, and I updated your guys' objective for today. We're kind of moving quick here. I also updated keywords. Remember, I told you there's a good amount of vocabulary in this chapter. Refer to your learning guide. I bolded the most important, if I remember right. I think I left Gene unbolded, and that would be ridiculous. So maybe circle or highlight Gene. That's very important. But anyway, the keywords, which one did I leave up there from last class? Which one did we deal with last class? Meiosis. So today is about probability, genotype, phenotype ratios. Okay, so this is a little bit of math here, figuring out probability. It's real easy math though, okay? So we use these Punnett squares. I'm going to teach you that or remind you of it. I bet a lot of you remember it from elementary school, or more importantly, or more commonly middle school. But anyway, I want to point out the word really describes it. Probably. Probability, probably what's going to happen. It does tell you all the possible options that can be inherited, and then it gives you the probability, the mathematics of what are the odds of it happening or not happening. So it's not for sure, just probably. Okay, so I think I kind of said all that. Genetic ratios are estimates of probability, not absolute numbers. I want to point out to you, though, there's many ways of recording the ratios or the percentages or the fractions, and you guys should be able to go back between them with no problem. For example, the most easiest one to understand is, you know, when your mom was pregnant with you and she didn't know if you were a boy or a girl, the probability that her baby was going to be a girl is what? Okay, so I heard a bunch of different ones. Awesome. So get used to using the percentage I heard. I heard a half, the fraction, which can also be in decimal, which is 0.5. And then more commonly we use ratios. And that would actually be a 1-to-1 ratio, like 50-50, right? So this is a ratio, so 1-to-1. So what it is you have one chance of getting the male, and you have one chance of getting the female. They should add up to all the possibilities, which is 2, okay, with genders for human beings. I'm recording this just so you know. Just because we want to hear you. Okay, so on to inheritance of alleles. So Mendel came up with 2 principles. Sometimes they're known as laws. But anyway, the principles of actually, that's kind of weird. In that case, 3. So this is actually not Mendel's law or principle or anything like that. It's just the principle of probability, I guess. So vocabulary first, and then I'll get back to what I was talking about. Monohybrid cross. What that means, mono means 1. So we're talking about one factor. So for example, we could talk about brown hair versus blonde hair. So it's hair color. Okay? Now, hybrid. Again, there are many words that are all going to mean the same thing in this unit, because hybrid means that it's a combination. So we would write that with big B for brown hair and a little b for blonde hair. That's a hybrid. It's also called a heterozygote. I think that's kind of it. The other one will have more options. I'll show you that in a minute. So we're going to cross basically a big B little b with a big B little b. That's a monohybrid cross. Now, the parental generation, the P generation, that's where we're starting. That's our first parents. The F1 generation is the offspring of the parental cross. The F2 generation is the offspring of crossing the F1 generation. So a little incest here. We're just going to cross these offspring to make the next generation. Okay? So just be kind of aware of that vocabulary. All right, is that familiar, you guys? Do you guys remember dominance and recessiveness? Okay, so there are many reasons why one allele is dominant over another. I'm not going to explain them right now. We'll get to that later. But just for right now, trust me, one allele gets expressed, the other one is silent. A lot of times it's simply a covering up. Like think of brown hair or black hair versus blonde hair. Okay, we'll talk more about that. So, one allele is dominant over the other. Dominance and recessiveness. And Gregor did figure that out. Okay, now, I think last class I might have told you that chapter 12 and chapter 13 are highly related, but at first glance, you might not see the connection. Here's the big connection. We did the dancing last time. What were we modeling? Meiosis. And meiosis shows Mendel's first principle of segregation. So, for example, this big B is on one chromosome. The little b is on another chromosome, right? We made these people. If I want to draw them into people, those are funny people. Okay? Anyway, they are going to segregate. You guys agree with me? Into what? This one big B is going to be into what? This little b is going to be into what? So when we danced and did the model, we ended up with 4 of these things. What were those 4 things? Gametes. They were haploid cells, daughter cells, not exact copies, half the information. Give me examples. Examples over here. What could this be? All right, what's that? Sperm. Okay, so see how they segregate. So this parent has 50-50 odds of giving away the big B or the little b. Okay? So we talked about that last time. We have to use this fact to make our Punnett squares. Already was. Perfect. Okay, now here's what I was getting at when I saw that word of principle. Mendel's first law or principle is called the principle of segregation. We did that when we did the model of the meiosis. These 2 alleles just segregated into their own gametes. That's simply all it is. 50-50 chance, right, of which allele moved into the gamete. And then that goes over it. Okay, this picture. Now, do you guys remember Punnett squares at all? So let's start from the beginning, because you might remember how to do this, but you're not understanding the true meaning. So at the very top here, we're going to cross our parental generation. We're going to go homozygous dominant with a homozygous recessive. And again, see the gametes? This one only can make one type of gamete. This one can only make one type of gamete, because that's all they had. They were homozygous, or true bred, or purebred. So see how there's more names for that one? And what happened is we put each parent on one side of the Punnett square. So do you guys see that on this Punnett square here? We segregate the alleles. Oops. We did quite a jump here. They didn't show you the Punnett square for this one. We'll do one of those later. So they're showing you the Punnett square for these parents here. Big R, little r, crossed with another big R, little r. And you guys remember how they come together? So big R, capitals first. Big R, little r. Big R, little r. Now, these phenotypes on this particular presentation have some typos. For example, Rough is dominant. Smooth is not dominant, but that sure looks like a rough P right there to me, doesn't it to you? So that should be a smooth guy right there. And then I think later they tell us somewhere that smooth is dominant, and that's not right. But don't worry, whenever we do them, the question will give you the information you need. So again, capital says it's dominant, lowercase says it's recessive. Okay, more vocabulary. I want you to memorize it right now, so focus with me. Genotype. See the word, the start of gene right there? So this is the actual genes. So this genotype, big B, little b, that's a genotype. It's the genes of the organism. In this case, it's a diploid organism, right? That's genotype. Now, here's examples. Big R, little r, and those are homozygous because they're the same. Then we get to our heterozygous genotype where they are different. Okay? So those are genotypes. What's the deal with phenotypes? I want you to memorize it right now. It starts with pH. I memorize this with physical characteristic. So what's the physical characteristic? Let's go back to my B's. What is the physical characteristic of the heterozygous genotype? Brown hair, right? What is the phenotype for big B? Brown hair. You have two possible different genotypes for one phenotype because of the dominance and when you have diploid organisms. The little b genotype was what phenotype? Blonde, beautiful. Okay, so there's Punnett square blown up a little bit, capitals first. You guys see where this is coming from? Here's a little one right here and a little one right here. Again, they've made a typo on the phenotype. Okay. Dihybrid cross. This gets a little more complicated. This is, not Darwin, this is Mendel's second principle. He figured out which is the law of independent assortment, or the principle of independent assortment. And this states that different traits or different alleles, like the blonde hair versus the brown hair, and then let's say another one like eye color, assort independently. This is the opposite of saying they're linked. Now, I started with that example because that does not follow the law of independent assortment. Eye color in humans and hair color in humans is linked. That means that it's found on the same chromosome. And it does have more likelihood of being inherited together. You guys can think about this. You know most people that have darker hair have darker eyes like me, right? Occasionally you will see somebody with dark hair that has light eyes, but that is rare. Do you guys agree with me on that? Do we have a couple in the classroom even? A couple in the classroom, okay? Only two out of, I can look around and we don't really have any true blondes in here, do we? So, and hair color and eye color is more complicated. It's more than one gene, okay? So that's why we get like dirty blondes, or we have the reds and all that. We'll talk more about that when you guys make your baby faces, okay? Now, Mendel didn't realize this, that there were these linked genes. Remember, he doesn't even know about chromosomes. He doesn't understand this at all. He is just, Curtis, you guys, he is just simply looking at the outcomes of his crosses and using that to figure out inheritance. So it's pretty amazing if you think about it. Okay, we have all this other knowledge now to explain it to us. But anyway, his principle here is the case in the pea plants. The traits that he chose to track happen to all be on different chromosomes. None of them were linked. You saw how we segregated into our gametes and our dancing. you guys didn't hang out together except for Lucera and Ray. They like to make sure that they were in the same half of the room or something, right? But so they were kind of linked, I guess. But normally, no linkage. Okay. All right. Now, to support this, he had to track 2 traits at one time. So that's what the di means in dihybrid. Once again, he was studying the hybrids to sort this. So you get this crazy Punnett square like that. Okay, now we will make one of these today. Now, something to realize here, as we're almost done with lecture, is this looks more difficult than it is when you make these. Now, you're going to get a Punnett square that already has the parent's possible gametes. Notice you... I'm going to